All right, so if you're logged on to this channel, it means you want to achieve great things in life. Welcome to Excellent Grades Academy. My name is Dr. Bison EM. Today we are looking at molecular biology. And in biology, we are focusing on nucleotides, a very high yield topic in UNZA exams. Recommend this video to all of your friends because this is very important. Very, very important. Without further ado, let's log into this video and learn. So our lesson objectives for today, number one, to describe the basic structure of nucleotides. Number two, to look at the functions of nucleotides and the therapeutic use of nucleic acids in medicine. Okay, so what you must know after this video is what nucleotides are, the functions of nucleic acids, and the composition of nucleic acids. This is very important and they will examine you on these facets. So everyone needs to make sure that they understand these things. Subscribe to our channel, hit that notification button for more videos like this. Okay, so a nucleotide is made up of three things. Okay. A nucleotide is made up of a nitrogenous base, a pentose sugar, and a phosphate group. What you must know is that a nucleotide is the building block of a nucleic acid. Our examples of nucleic acids are DNA, here where the cursor is, which is called deoxyribonucleic acid, and RNA, which are your ribonucleic acids. So nucleotides build up our genetic material, which is your DNA and your RNA. There's going to be a video where we'll be looking at DNA and RNA and their characteristics. Okay, so nucleotides are made up of three things. They're made up of a nitrogenous base, this substance that is in green here, a pentose sugar, this that is in blue, and a phosphate group, this that is in pink here. Okay, now if you have a substance that is only made up of a nitrogenous base and a pentose sugar, that is called a nucleoside. So the difference between a nucleotide and a nucleoside is that a nucleoside does not have a phosphate group, while a nucleotide has a phosphate group. Just to recap, a nucleoside is just made up of a nitrogenous base and a phosphate group, while a nucleotide is made up of a nitrogenous base, a pentose sugar, and a phosphate group. All right, so to further go deep, this is the structure of a nucleotide. As you can see here, this is a sugar. This sugar has got carbon 1, 2, 3, 4, 5. So because it has 5 carbons, it is called a pentose sugar. And a nitrogenous base here is attached to carbon number 1 of the pentose sugar. This bond here is called an N-glycosidic covalent bond. So the bond between the pentose sugar and the nitrogenous base is called the N-glycosidic covalent bond. And then you have on carbon number 5, a phosphate group is attached, okay, through a covalent bond as well. This is a phosphoester bond. It's a phosphoester bond because, as you can see here, this is a phosphate, this is oxygen. So this is a phosphoester bond. So a phosphate group is bonded to carbon number 5 of a pentose sugar through a phosphoester bond. So now, let's go deep into this and explain the individual components of a nucleotide. All right, so nucleotides are made up of nitrogenous bases. Nitrogenous bases, what we saw here, this is a nitrogenous base. Now, a nitrogenous base is simply a base that is cyclic and contains nitrogen. There are two types of nitrogenous bases. We have what we call purines and pyrimidines. So everyone needs to understand what a purine is and what a pyrimidine is. A purine is a 9-membered double-ringed nitrogenous base. So a purine just has two rings. A purine has two rings, whilst a pyrimidine has got one ring. There are two examples of purines. You've got adenine and guanine. So, listen to this. Purines have got two rings. And there are two examples, as we are going to see as this video goes on. There are two examples of purines, which are your adenine and your guanine. You can even see from the name themselves, the guanine and the adenine, that they have 9 carbons in there. Adenine, guanine. Pyrimidines have got six atoms in a ring. They only contain one ring. So these are 6-membered heterocyclic aromatic rings. Examples of pyrimidines include uracil and the other examples that we are going to see. Okay? And the other examples that we're going to see: uracil, thymidine, and cytosine. Okay, so we're going to, when we start doing nucleic acid, we're going to see which type of pyrimidine is exclusive to RNA and which one is exclusive to DNA. Let's look at these structures. So these are your nitrogenous bases. On top here, these ones that have got two rings, as you can see, you have a ring here and another ring here. These are purines. There are two examples. You've got adenine and guanine. You've got adenine and guanine. One of the most visible differentiating features between adenine and guanine is that guanine has got a double bond to oxygen here on carbon number 6. It has a double bond to oxygen on carbon number 6, whilst adenine has got a double bond to an amino group. This is called an amino group. All right. Now let's move on to pyrimidines. Pyrimidines have got one ring. They are called monocyclic. They are monocyclic because they have one ring. Our examples are uracil, this one here, cytosine, and thymine. So cytosine is found both in DNA and RNA, whilst uracil is only found in RNA. Uracil is only found in RNA, thymine is only found in DNA. So your pyrimidines are monocyclic, whilst your purines are bicyclic. That means they contain two rings. Okay. So those were our nitrogenous bases. Another components of nucleotides are your pentose sugars. Now, pentose sugars are known as pentose sugars because they contain 5 carbons. They contain 5 atoms in their rings. 5 atoms in their rings. Because they contain 5 atoms in their rings, they are known as furanose 5-membered rings. They're known as furanose rings. They're known as furanose rings. There are 2 types of pentose sugars. There is what we call a ribose sugar and a deoxyribose sugar. The difference between a ribose sugar and a deoxyribose sugar is that a ribose sugar has got an OH on carbon number 2 and a deoxyribose sugar doesn't have an OH on carbon number 2. Let me just repeat that because that is a very important point. A ribose sugar has got an OH on carbon number 2, whilst a deoxyribose sugar does not have an OH on carbon number 2. Furthermore, ribose sugars are found in RNA, whilst deoxyribose sugars are found in DNA. So that's why your RNA is called ribonucleotide. It is your RNA is called your ribonucleic acid because of the ribose sugar, whilst your DNA is known as deoxyribonucleic acid because of your deoxyribose sugar. So that's the main difference between your pentose sugar and your that's the main difference between your ribose sugar and your deoxyribose sugar. So you can tell actually that this is a ribonucleotide or a deoxyribonucleotide, or whether a structure is DNA or RNA, by the type of sugar that they contain. If they contain ribose sugars, they are RNA. If they contain deoxyribose sugar, they are DNA. Okay, so this is a ribose sugar here. This is a Fisher projection, and then this is the ring structure. It is a furanose form. Okay, this is a Fisher projection of your ribose sugar. This is your ring structure, also known as your Haworth conformation. And then this is your Fisher projection of your deoxyribose sugar, and this is its Haworth projection. Now, I want you to notice on carbon number 2 here, on the ribose sugar, you find an OH. But on the deoxyribose sugar on carbon number 2, you don't find an OH, you only find an H. That's why this is called deoxyribose. Why? Because an oxygen is removed at carbon number 2. So it is deoxyribose. Deoxy simply means remove oxygen. So they removed oxygen at carbon number 2. That's why it is deoxyribose. Beautiful. The other component and the last component of a nucleotide is a phosphate group. It is a phosphate group. So a phosphate group is simply a phosphate that is double bonded to one oxygen atom, and it has 4 hydroxyl groups single bonded to it. So the phosphate group is usually bonded to carbon number 5 of the sugar. And remember, when we were beginning this, we said that the bond between carbon number 5 of the sugar and the phosphate is called the phosphoester bond. The phosphoester bond. So when you have a nitrogenous base that is bonded to carbon number 1 of your pentose sugar, and you have a phosphate group that is bonded to your 5th carbon of your pentose sugar, then you have a nucleotide. All right. So this slide is just showing us examples of the nucleotides that we have. Now let's look at this. If the nitrogenous base that is bonded at carbon number 1 is adenine, and there's only one phosphate group that is bonded to carbon number 5, the name of this nucleotide is adenosine monophosphate. It is adenosine monophosphate. Sometimes we will also include the number where the phosphate is bonded. So this can be adenosine 5' monophosphate. So carbon number 1 is called 1'. Carbon number 2 is 2'. Carbon number 3, 3'. Carbon number 4, 4'. Carbon number 5, 5'. Just like that. That's how we name our carbons. All right. Now, let's look at how these nucleotides join together to form our nucleic acids. So what happens is that for nucleotides to form nucleic acids, if this is nucleotide number 1 here, adenosine monophosphate, and this is nucleotide number 2, adenosine diphosphate. If we want this nucleotide and this nucleotide to bond together to form a chain, the phosphate group of nucleotide number 2 is going to bond at carbon number 3 of the sugar of nucleotide number 1. Okay, when there is bond, these two nucleotides will be joined together in a bond that we call a phosphodiester bond. So nucleotides bond together to form a chain known as a nucleic acid through a bond called a phosphodiester bond. That is different from what we were talking about, a phosphoester bond. A phosphoester bond is formed when A phosphate group is bonded to the 5th carbon of the sugar. That's what we call a phosphoester bond. That is a phosphoester bond. But when we bond two nucleotides together, look at this. This is nucleotide number 1, nucleotide number 2. When these two nucleotides bond together, they form this bond here. This bond is known as a phosphoester bond. A phosphodiester bond rather. It's known as a phosphodiester bond or a phosphodiester linkage. A phosphodiester linkage or bond links nucleotides together in a chain, and this chain is what we call a nucleic acid. So a chain of nucleotides is what we call a nucleic acids. Remember in the beginning, we said that nucleotides are building blocks of nucleic acids. So nucleic acids are just polynucleotides. Polynucleotides, meaning nucleic acids are just chains of nucleotides that are joined together through a phosphodiester linkage. Okay, so I want you to note the sugars that are present in this chain here. Look, this sugar doesn't have an OH at carbon number 2. So this is a deoxyribose sugar, meaning that this chain here is DNA. But when you focus on this chain on this right side, on carbon number 2 you have an oxygen, meaning this sugar here is a ribose sugar. Since this is a ribose sugar, this chain is RNA. That's how you differentiate. Okay. A glycosidic bond, we said, is simply a bond that joins the nitrogenous base and the pentose sugar here. So it is the nitrogenous base is joined at carbon number 1 of the pentose sugar. And what happens is that the bond is between the carbon and the nitrogen. This bond is covalent in nature, but because it is between the nitrogen and the sugar, it is known as a beta-N glycosidic bond, an N glycosidic bond, an N glycosidic bond. The reason why we call it beta is because the nitrogenous bond is on top of the sugar and the H here is below. Okay, so as you can notice, the nitrogenous base is on top of the sugar. So because it's on the top position, this top position is called a beta position. So that's why this bond is called a beta-N glycosidic bond. Beta-N glycosidic bond. All right. So these are just examples of deoxyribonucleotides that can form. You've got this one. So this one has got adenine as a nitrogenous base. So it is known as a deoxyadenosine 5' monophosphate. This one has got guanine as a nitrogenous base. So it is a deoxyguanylate, deoxyguanosine 5' monophosphate. This one here has got thymine, so it is deoxythymidine 5' monophosphate. This one has got cytosine as a base, nitrogenous base. So this nucleotide is known as deoxycytidine 5' monophosphate. So you name your nucleotides depending on what type of nitrogenous base they contain. All right. Now, let's move on. What are the biological importance? What is the biological importance of nucleotides? So, those nucleotides that contain adenosine, number 1, can serve as an energy source, okay, because they form a molecule that we call ATP. ATP is needed for energy for muscle contraction. So, for you to contract your muscles, you use a nucleotide known as ATP. ATP is simply adenosine triphosphate. So meaning this is just a nucleotide that contains 3 phosphates. Adenosine triphosphate. It is used for provision of energy used for muscle contraction, to transmit nerve impulses and for your sperm to move. Spermatozoa motility. In order for your sperm to move, they need energy through ATP. Okay? ATP or adenosine containing nucleotides are also required for activation of methionine formation in the methylation process. It also plays a role in peptide and protein synthesis from amino acid. Okay, ATP is also converted to ADP and AMP in vivo. Those nucleotides that contain guanosine play an important role in protein synthesis, just in the formation of proteins. They play an important role in the formation of protein synthesis. When you do translation, you're going to notice that for you to activate an amino acid so that you can form a protein, GTP, which is known as guanosine triphosphate, is utilized. Okay, so it also takes part in purine synthesis and is also necessary for gluconeogenesis, which is one of the processes in the liver that gives you glucose. All right, so as we conclude this video, I want you to take note of this. Ribonucleotides are used to synthesize RNA, and deoxyribonucleotides are used to synthesize DNA. For further reading, you can read more on the use of nucleoside analogues as therapeutics, which is medicine for cancer, and read more on HIV biochemistry, read more on the base of the listed approved drugs that are used in HIV. This is Dr. Bison EM. You may join Excellent Grades Academy and register for lessons in biology, in mathematics, in physics, and in chemistry on the number that is projecting on the screen. Please subscribe to this channel as we will be doing videos in all the courses. Study hard. Your future is as bright as the sun. See you in the next video.